And I've been working with a group of faculty colleagues here. Let me introduce Dr. Charlie Goldberg, who's part of the faculty committee and who's going to be the emceeing the morning here.
Okay.
Charlie.
Okay. This on?
Yeah.
So, yeah, great. Okay, welcome everyone. Day 2 of our symposium. I suspect people will continue to dribble in for the next hour or so. Those who were here yesterday exceeded their expectations in terms of attendance, and the content was fabulous. A lot of great speakers, and we have additionally great speakers today. I want to acknowledge Ms. Christian for putting this all together and being the driving force. And without it, we wouldn't be able to have this. The whole point is really to make the campus smaller. We have so many great resources and people around here, and they're often unknown to each other. So in health sciences, I'm the Associate Dean for Graduate Medical Education, which oversees our residents and fellows. We have 150 programs and 1,100 trainees. We have 1,700 faculty. We encounter challenges all the time, and people often get frustrated and don't take it to the next step of solving the problems because they don't know who to reach out to. They don't know the methodology, and that's a shame. So this place is very rich. I've been here for 26 years, and I'm constantly amazed about how welcoming everyone is in engineering, in business, in social sciences. They want into the health system space, and we need everyone's help. So by having these kinds of sessions, we help to lower the activation energy that's necessary to get in, to solve problems, often to create companies and to innovate, and to deliver better healthcare. So with that, this morning we have a number of great speakers talking across a range of topics. First, I'm pleased to introduce Dr. Kit Poliano, who is the Dean of Biosciences, and she's going to talk about commercialization of new technologies and tell her story. A lot of this about hearing stories. Kit.
Thank you so much. Let's see, I'm gonna put this here and see if it.
Yeah.
Okay, all right, can you hear me?
Yes.
A little closer.
A little closer, okay, there we go, right up there. Okay, well, thank you everyone, and happy Saturday. I hope everyone's doing well. So I'm gonna talk about a journey that really was a partnership with myself and my husband, Joe Poliano, who's here in the audience and who will join in the Q&A at the end of the talk. So please keep in mind, everything that we did here was a true partnership. So I'll talk a little bit first about the basic need for new antibacterial ways to treat drug-resistant bacterial infections, and then dive into our specific launch story. And first, I'm sure everyone in this audience is aware that we're losing the evolutionary race with antibiotic resistant bacterial infections. The number of resistant bacterial infections continues to climb, while the number of new FDA-approved antibiotics continues to decline. And this is a global problem with 1.27 million people killed per year in 2019, and associated with, 5 million deaths. The CDC watch list has this wide range of microbes, and actually there's more being added almost every day with serious threats, not only from drug-resistant bacterial infections, but also fungal infections that can be really quite challenging to treat. And one notable infectious disease is tuberculosis, which infects nearly 25% of the world's population, causing a remarkable 3,500 deaths per day. Highly drug-resistant TB strains are emerging and can require many months to years to treat. Treatment success declines with increasing antibiotic resistance, and per year there are more than 400,000 resistant infections and 1.3 million deaths from tuberculosis. So it's really quite a pressing global challenge. And most concerning of all is that the future looks not very good at all, with deaths from resistant infections predicted to equal cancer by 2050. And there's a fabulous new report from the UN Environmental Program on this challenge that I encourage you to take a look at. And from an innovation perspective, few new antibiotics have been introduced, and when new drugs are deployed into the clinic, resistance immediately arises because there's a huge selective pressure and there's large numbers of microbes in the patients and in the environment. And so it makes it pretty easy for resistance to emerge. So Joe and I started this company from our academic foundations. I am a microbiologist and I study bacterial cells and developed many live cell ways to study how bacteria do special things. Like in this case, this time lapse of the spiral is really a time lapse showing bacteria making endospores. This is Bacillus subtilis, but the same thing happens with C. diff and other really nasty infections, anthrax, botulinum, are all caused by these spores. And Joe is also a professor at UC San Diego, and he discovered these bacterial viruses that assemble this amazing nucleus-like structure in bacterial cells, basically converting a bacterium to a classical eukaryotic cell structure. And in 2011, we took a joint sabbatical, much to the horror of our students and postdocs. We started working in the lab, which, they're pretty sure we forgot everything we knew about how to work in a lab at that point. And we jointly discovered this new approach that we call bacterial cytological profiling. And this is an image of an E. coli cell that looks happy and healthy, and the red's the membrane and the blue's the DNA. And I think you can tell that in these three images on the right-hand side of the slide, that those cells look really different. And these are cells that have been treated with antibiotics. And then this is generally applicable, and you can get all of these different phenotypes that really correspond with inhibition of different pathways. And so we called this method bacterial cytological profiling, or BCP. And we found it to be just really amazing at pinpointing not only the pathway that was inhibited, but in many cases the step in the pathway that was inhibited. This is really perplexing. If this is a eukaryotic cell, I don't think it would work quite as well, although I'll show you later that it does work in some eukaryotes. And so then after this, we measure cell parameters, and then we do mathematical analyses, starting really with PCA and LBA, but moving now into AI and machine learning approaches. We were really excited about this because mechanism of action determination is a key rate-limiting step in drug discovery, and in antibacterial drug discovery especially. And traditional approaches require months to years, and they're very expensive, and they often fail. And we felt BCP was a solution to this. So in 2011, we filed a UC San Diego disclosure and started going down this patenting aisle. And we were enthused about the benefits of this. BCP is really rapid. In basically a day, you can get insight into the mechanism of action of a drug, which is unprecedented speed. It's versatile, works for all drugs, all organisms, and it's really reliable, at least in the right hands. And it even works for really hard to characterize membrane targets, which are super challenging. The drawbacks are that BCP requires really specialized experience, and technical skills. So you had to be able to grow bacterial cells and keep them alive and keep them looking healthy. And Joe and I can tell you that if you look in the literature, many papers have images of dead cells that are thought to be live cells. So if that's the case, you can't use this method. It requires costly instrumentation and lots of comparison data for different organisms and different drugs. And the more experience and the more data you have, the better the method is. So we started trying to license the technology and had challenges getting the license, getting other companies to pick up the license. And that was because of these drawbacks, really. It wasn't really quite ready to be just shipped off there into the wild world of commercialization. And so we did two things, really. First, we continued to validate the technology in our academic labs. And we have more than 60 co-authored papers using BCP technology with a growing number of citations, more than 20 million in funding. And we were really happy when, in 2017, so 5 years later, we won the O'Meara Prize for Best Original Article in the Journal of Antibiotics and the Editor Spotlight selection. And we were happy about that because we were not from the antibiotic field. And this showed that we were finally accepted by the antibiotic field. And that not being a part of the club was part of the reason we couldn't license the technology also. We were these, faculty, we have this great technology, we think it's the thing to solve all problems, right? But now we were a part of the club and it was really validated. So the two keys, the people who really launched this in our labs are Ann Lamsa and Mike Nanajui. They were fabulous students in our labs, published really key papers, including the two founding papers of the technology and the one that won the Young Europe Prize, which is this one here. Really happy with that. And then basically at the same time, we decided to co-found Linnaeus Bioscience Incorporated to take this technology out from UC San Diego. And so we licensed the technology back from the university and bootstrapped the company. So we only had our own modest investment to get the company going. Our first phase was in JLabs, but actually before we were in JLabs, and actually continuing after, we were also doing experiments, and we really means in this case Joe, in using university facilities using that external rate for companies. So we really were hatched here at UCSD in some ways, in many ways. And we've been profitable since day one. And one of the things we were really lucky about was that we were able to hire my very first PhD student, Mark Sharp, who went on to get his JD from UC Berkeley's famous School of Law. And he became our chief scientific officer. And he was actually also able to help us review contracts and things as well, although we did hire outside legal counsel. He was a really key person. When he was in my lab, he was one of these people that could get anything to work. Like just if you had some challenging technique nobody else could get to work, well, he knew Mark could get it to work. So that was really, really wonderful. We have two business units. First is the CRO to provide mechanism of action work globally to companies, and our first client was Cubist Pharmaceuticals. They're the ones who allowed us to rent that space in JLabs and others that came along. And then we also have a pipeline program to identify novel antibacterial drugs. And this we do via corporate partnerships with companies that have chemical libraries. And we've developed some partnership models that we can talk about later. Right. And the Linnaeus BCP platform looks a lot like the ones we developed in our lab many years ago, although it's diverged, of course. And one of the challenges is keeping these two things really separate. That's an interesting challenge. One of the things that we've added to the Linnaeus platform is a proprietary AI platform to analyze the data sets. And this is Joe Sugi is our computational biologist. And the advantages, of course, is that you can, AI and machine learning can allow you to better recognize and classify shapes. It's automated, much faster. No human expertise is needed. So gone are the days when Joe and I would be peering over somebody's shoulder saying, what does this look like? Does it look more like this or like that? And the power increases with increasing data size. So the longer the company's in business, the more reliable the tool gets. And it can be applied to any cell type and any kind of type of thing that kills bacteria. We have 11 years in the CRO business. We're a global leader in MOA determination, custom cell biology, and microbiology. So one of the things the company provides is microbiological expertise and consulting. So basically with the contract typically comes some consulting as well. More than 35 clients, many repeat in North America, Europe, Asia, and Australia since 2012. And key to this is Hannah Tsunemoto, who is our Chief Operations Officer. She's a really talented former PhD student in Joe's lab who was hired to the company right after she got her PhD. We also have research and development collaborations focused on MOA determination for different kinds of organisms and drug discovery, including antifungal drug discovery and, interestingly, hepatitis B capsid inhibitors and tuberculosis. We're the global service provider for the TB Alliance and developed the BCP AI platform with Gates Foundation support. And so what this means is that the company receives molecules from all the different members of the TB Alliance and provides mechanism of action insight into how these work. And we tested more than 100 compounds in 2023. And we're also identifying new compounds that are active against extremely drug-resistant tuberculosis. We also have relied upon, in addition to the contract work, extramural funding from not only the Gates Foundation, but also the National Institute of Health. And this allowed us to further refine the platform and apply it in drug discovery efforts. And one of the advantages to BCP is really, if you compare it to a traditional phenotypic screen, You might get 1,000 hits that kill your target organism, and then typically you filter these out by things that are really independent of mechanism of action, and you might end up with 10 that you take into further development. And therefore you throw out the vast majority of your hits, and any new mechanism of action will go in the garbage along with, yeah. In VCP, in contrast, you can get the mechanism of action for all of the compounds that are hits and throw away the unwanted mechanism of actions, and then just have a few targeted mechanism of actions that you put into the development pipeline. And this is really advantageous. And we have our internal pipeline. We have 3 different main programs that we're in the process of taking through in SAR and lead detection, or lead selection and optimization. So I wanted to turn next to the business of antibacterial drug discovery. And I think the bottom line is that Big Pharma ditches antibiotic discovery. In 2015, our very first client was purchased by Merck. This is Cubist, who brought daptomycin to the market. That's a big drug, right? I think last I knew it was about $1 billion a year. Is that right, Joe? Joe says, yeah. Still $1 billion a year. But then they closed down their discovery pipeline. They had other drugs in the pipeline, and that was all gone. This was followed by AstraZeneca spinning off their anti-infectives research, and Sanofi transferring their infectious disease unit. Novartis completely dropping their antibiotic drug discovery push in 2018, and then just last year, J&J ending R&D, not only for anti-infectious drugs, but also vaccine development. It's a little bit alarming. And so I thought it would be interesting to dive into one especially notable collapse in this industry. And this is Achaogen. And Achaogen was a company that developed plazomicin, which is a novel aminoglycoside with activity against DRE. These are infections that cause a lot of problems in the clinic, as many of you probably know better than I. They had positive results in a phase 3 trial comparing it to colistin, which is a really nasty drug, but is a drug of last resort for many infections. They received FDA approval for complicated urinary tract infections in 2018, and in that year had $800,000 in annual sales. Seems pretty good. Well, what are the challenges? Well, FDA rejected their application for use in sepsis, which is a nice market that they were counting on. They had their first clinical trial in 2018, and it took them 9 more years to get this thing licensed by the FDA. And they spent $660 million in R&D and post-approval steps. The rarity of AMR infections made it really hard for them to conduct a clinical trial, which was very expensive. Screened 2,100 patients just to enroll 37, because thanks to all the good work in the clinic, these infections are really low. We need to keep them low. But then how do you do trials to show that your drug outperforms the last ditch drug, colistin? Very challenging. The final challenge is perhaps the biggest of all, and that is when you develop a new drug, you have to reserve it for only those few cases where you truly, really need to use it, because otherwise resistance will evolve in the clinic. And so their total sales were just $1 million after spending $660 million. So I guess it's no surprise, it was sad to see them declare bankruptcy in April 2019. So I guess the key lesson is that the antibiotic business model is broken. It typically costs $1 billion to develop a new antibiotic, and only 1 in 30 new molecules that are developed will actually make it into clinical trials. And we need to limit sales, so your annual sales will likely be around 100 million. So it takes you 10 years to break even. And there's a really wonderful article, recent article in Nature about this. If you'd like to read about this, it's a fabulous segue into more literature on the topic. So what are the solutions that we can imagine in this space? I think first, push incentives to fund and reduce the cost of early stage drug discovery are critical. We've benefited from those push in Linnaeus Bioscience. NIH and CARB-X and Enable are really important. And consortia as well are really important to reduce the risk and the cost and really hedge your bets and have lots and lots of people discovering drugs. And we also view Linnaeus as a consortia. So we wanted this technology to get out there and benefit as many companies as possible. And so we view our role as the company is really to accelerate antibacterial drug discovery by helping as many people as possible who have a new molecule that they think might be useful. If only 1 in 60 will make it into the clinic, we need to screen as many molecules as possible and help companies out. I think we also need pull incentives to address the market failure, because the market will always be challenging for antibacterials because we have to reduce the emergence of resistance. And there are a few different approaches have been suggested. One are guaranteeing a minimum purchase amount from the governments. Others are rewarding companies when they bring new drugs to market. And third, a subscription to pay companies based on the drug's value rather than the sales. So kind of switching this around and viewing this as a public health threat and therefore finding new ways to invest and make it. Sustaining innovation is absolutely critical in this space. So we need as many new companies as possible in this space to have, so we maximize individual creativity and innovation. We critically need innovation to meet this. The small companies in academia can also provide the sustained focus that clearly is hard to maintain in big industry. And for this, we critically need funding from federal agencies and private foundations. It's really important. Okay, so I wanted to spend just the last couple of minutes talking about two approaches from our science. So these are things that are currently still at UC San Diego, well, more or less, and developing, and one is personalized medicine to really tailor the antibiotic to the patient. And the second is developing more effective phage therapy for personalized medicine. So first, Diana Quach was our joint PhD student who graduated in 2016. And in her thesis work, she found that BCP could serve as a rapid diagnostic, and therefore you could screen patient samples against a panel of different antibiotics and identify the best antibiotic to treat the patient. And this work, Diana's work was a collaboration with Victor Nizet and George Sekoulas, two MDs in the School of Medicine who are probably well known to this audience. And so we have some nice publications there. This is something that really hasn't been scaled, but is definitely scalable with current technology. And the second I wanted to talk about is phage therapy, which is, well, weaponizing natural predators of microbes. So almost everything on Earth is infected by by many viruses. And this includes bacteria. And bacteria infect viruses, or phage infect bacteria, and then the bacterial lice. And these can be used to treat antibiotic-resistant infections. And it's an exciting new front in personalized medicine. And in fact, at UC San Diego, we have the IPATH Center, which was founded by Chip Schooley and Steph Strathdee. And this is the first dedicated phage therapy center in North America. And their mission is to bring innovative research and clinical practice to the field of medicine. And it's the story of the founding of IPATH is really fantastic, and they're just two dynamite leaders on the campus and in this field. And as I mentioned earlier in my talk, Joe discovered this special family of phage. And these are phage that are called jumbo phage. And here is one of the classic phage, T4. And you can see that the jumbo phage are much larger than this phage. And on the infected bacteria here, their DNA ends up packaged in this structure that looks just like a nucleus. And what that does is it protects the DNA from all DNA-based bacterial immunity systems, restriction enzymes, CRISPR, all of these many different things. And so we thought that these would be fabulous tools for phage therapy. They're broadly resistant to DNA-targeting bacterial immune systems. They have a large genome, so we can remove a large chunk of it and add a payload to it. And they're less likely to transfer virulence genes. So I don't know if you know, when phage infect bacteria, they can pick up the bacterial chromosome. But in these organisms, the phage chromosome is separated from the bacterial chromosome. So there's no way you're going to transfer the cholera toxin gene to a different species, for example, or a different organism. And discovery of these, I wanted to just point out, these depended on a collaboration with Elizabeth Villa, a faculty member at UCSD, who developed this fabulous approach for looking at bacterial cells. And here in blue, you can see the phage nucleus. In green, you can see the assembling phage capsids. And in yellow are all of the ribosomes, which, just like in a eukaryotic cell, are excluded from where the phage DNA is. It's really quite remarkable and a fabulous collaboration with Elizabeth Steinmeier. So what is the jumbophage project? So the project is to identify minimum genome, build a minimal genome phage, add payloads, antibiotics, countermeasures to the bacterial immune system, and things that might help them recognize a variety of different host bacteria. And then to evolve them so that they recognize a diverse host recognition, they recognize the right species. And this is a fabulous project. This is one of a really dynamic team of researchers and funded by the HHMI. And it includes both IPATH and the Yale phage therapy team. And we have biochemists, structural biologists, cell biologists, geneticists, and two individuals on the clinical and phage therapy side. So it's a really wonderful, wonderful team. Okay, so to close, I just wanted to say that antimicrobial resistance is a classic One Health topic, and drug-resistant microbes spread between animals, humans, and the environment, of course. And the climate change, biodiversity loss, and pollution will increase the toll. And innovation, therefore, must accelerate and extend not only from healthcare and drug discovery, but also to agriculture and environmental health. So it's really important that we sustain innovation in these areas. I'm ending on a different point than launching a company, but anyway, I wanted happy to answer questions at this point, and I'll ask Joe to join me if you have questions. And thank you for your attention. Yeah, Corey.
Hi, amazing story at work. This is really cool. One thing, as someone who has never started a company but has a couple patents, including on software and on drugs, I wonder, how do you balance the mission of academia to make our discoveries open source and available to the public versus the need to keep things proprietary for your companies?
Yeah, that's a dynamite question. So I guess we should start. I'll start, and then I'll let Joe fill in. First, our patent was never approved. Okay, so we didn't have a patent. So our entire company is based on trade secrets and our reputation in the field, right? So it's yeah. So that's one piece. Second, at the time when we both had the company and were publishing papers, we had to go through all of these campus regulations, right? The conflict of interest, conflict of commitment had to be reviewed every year, and we had to have disclosures in all of our presentations and papers. Yeah, super. But we're now past that. Joe, maybe do you want to fill in a little bit more?
Yeah, what I would say is there's an absolute need for both. You need academia, you need publishing, you need no IP and getting information out there. On the other hand, if you have something like a drug, a molecule that you think has potential, if someone doesn't own it, then it can't be commercialized.
That's true. That's true.
So you have to have, for those select projects, you have to those are the molecules or projects that are ripe for basically spinning out.
To be clear, I'm not arguing against any of that.
Yeah.
I'm just wondering, so for example, I have funding to make open source software. And my funding is to make it free
Yeah.
and open. So how do I decide, like when it gets down to the details, like what am I going to keep for a company versus what am I going to publish and make open source, and how do I maintain my reputation as an open source scientist while also having closed proprietary stuff?
Yeah, boy, I mean, this is especially as we move into the AI world where the training data sets get more and more valuable. This is a huge question, I think, for all of us. I think the university has a special role to play in this world, at least in the foundational biology and biomedical research world, because our scientific expertise on the campus is really great. We can make sure those training data sets are good data sets and don't, in our case, include dead bacterial cells, which is what would happen if you just perused public data. I'm not quite fully answering the question. Maybe we should turn to this at a discussion at the end.
Yeah, I see Chris, you're kind of jumping. If you want to take that on, Chris Lorman, actually, this is his area of expertise.
So hi everyone, I'm Chris Lorman. I'm Associate Director for Life Science Licensing. It's a spectrum. There's not one answer. I'm very proud to say here at UC San Diego, thank you, we patent-enabled control. We patent things and to be able to give them away. Because if we don't patent it, somebody else might, or they'll put a ring fence around it. A good example, it was Zika. We had a technology there, well, actually, that's exceptional. I worked with the faculty, we positively decided not to patent because the patent wouldn't matter, it was an emergency situation. In other instances, we patent in order to control it and be able to give it away potentially, and that's our choice. And I will say that we, I hope, never interrupt faculty publication. We work with them. We put in place the IP protections to enable some control. Remember, these technologies, any of you who live in California, it's your property. We just take care of it on your behalf. And sometimes with that control, we can choose to give things away. So it's a balance, but then we're always trying to work with our faculty to make sure they can publish very, very quickly. Happy to talk more.
Hi. My name is Ajay Bharti, and I've been practicing infectious diseases here at UCSD for 2 decades now, and also doing research in malaria, HIV, TB. So I'm very excited to hear this talk on antimicrobial resistance and ways to overcome this. Antimicrobial resistance is described as a silent pandemic, and we're all familiar with pandemics now. But it's just a comment. I think you very well highlighted the issues, ranging from the financial aspect where the big pharma companies cannot make money from this and are not interested. But something needs to be done, and using AI and other technology, I think this will be very helpful.
Thank you.
Hi, my name's Alan. I'm one clinician. Just a quick question. In the project where you're using the platform to guide antibiotic therapy, what is the benefits of this platform compared to what we're already using? What's the turnaround time?
Do you want to do that?
Sure. The benefit is that once you have the organism, that you can, within a half hour to an hour, immediately get a signature and a response from the bacteria sensitive or resistant. You can do it in high throughput, really fast. As opposed to waiting, if you're just doing the traditional test, you wait 24 hours frequently. So this gives you much faster. In theory, you could pull individual cells out of a patient on therapy and look at them and say the therapy is effective. So it could be really great.
Thank you for the question.
That's a really good technology. What's stopping it from scaling?
Oh, Helen, back to you.
So we've been reasonably successful with LMAS in terms of our people service, et cetera. If every time when we go and we talk to somebody who might want to invest in a diagnostic, their eyes glaze over. They're worried about the margin, they worry they can't make enough money, they worry it's too challenging. And we just haven't been able to get any significant investment to roll this out. We're gonna try it again this year.
Chris. Thank you for that excellent talk, Dean. So I'll just say that this is a dream for us to see a faculty-initiated company that licenses, goes on and gets SBIR. This is an ideal pathway forward. I wonder, could you give any feedback, any thoughts about the process around licensing and also the SBIR and the journey into growing your company?
Yeah, thank you. Thank you. Maybe I'll, the licensing, first this happened in 2011 when we filed the disclosure of the licensing in 2012. So that was the ancient era. And, we did have issues, because of the way the terms were set up at that time. I think now it would be much more straightforward. Do you want to answer the other piece or say anything else about that?
Well, I think the original licensing was pretty aggressive, and I think things are much better now. And then in terms of growing the company in SBIR, you know, I guess maybe one of the goals of this is to give people like ideas of how to get started. And so I'll just say that if you're a faculty member out there and you have some ideas, you think, I think I have maybe already a patented molecule or technology, the first thing everybody told us to do is talk to other experts in the field, especially experts in industry. And if possible, go to the meetings, not just with your academic friends, but where the business people go. And start learning about your business side and how they would view the technology. In our case, there was a meeting on antibiotic discovery that was half academic and half industry. And so I would go to that meeting, I would meet all these people, the leading experts, they've been doing antibiotic discovery for 20, 30, some for 40 years, and I could get their feedback. And fortunately, they said, yeah, this looks really good. In fact, it looks so good. If you found a company, we'll give you a contract tomorrow, and that's how we got going. I think talking to experts. Another thing people told us is start writing SBIR grants now or other grants now if those grants are available for your technology. Because people say, oh wow, gee, it takes so long to get a grant. It's like, well, the time's going to go by, and you're still going to need 500,000. You can do a lot with 500,000 or even 300,000. So you've seen, we've written a lot of grants. Those grants were actually critical in getting us going and maintaining us in the early years, especially. So, yeah, and so talk to as many people as you can. And if, you know, this campus has had a lot of very, very successful alumni. Many of them, some of them are VCs, many of them are entrepreneurs, and talk to them and get their advice. And that's something we did. We talked to a former PhD grad student who was in venture capital for a decade, and he was super helpful. He said, don't take VC if you don't.
It's great that you share your origin story, and I think it's neat that you're a couple too, and you kept that together as you've grown your companies. When you talk about finding other people on campus, because that can be daunting when people live in silos, how did you go about doing that?
You know, we talked to faculty that we knew had startup companies. You know, we knew, you know, I knew in biology who to go talk to. At that time, there weren't that many. So that was one way we went about it.
Yeah, talk to your colleagues who started companies as well. And everybody will be super generous and supportive. We've never met one person who just said, go away. Everybody wanted to share their experience and give advice.
This is a very technical question, but I've only written R01s. I have no idea what writing an SBIR is like. Is it harder? Is it easier? Is it more of one or another? And you talk about like the time that it takes to do an SBIR versus an R01.
You know, it's typically a little bit smaller, it's extremely focused. If you've got a new technology that's exciting, our first couple of SBIRs were funded right off the bat. Program officers are fighting over the chance they have in their portfolio.
But it's like you need to have a company already, right? You have to form the company.
Yeah, but forming the company is easy. People can help you with that.
Yeah.
We have training course on doing SBIR applications in this building, so please join us.
All right.
And to begin the process, then you don't need help.
The NIH also has a training course that's available. They'll actually assign you somebody who will go through the grant process with you. But if you're regularly doing NIH grants, an SBIR will be a breeze.
Oh boy.
And I think, I mean, you're also a nice example of the crossover between basic science folks and working with clinicians. And as a clinician, is there an approach that you took to engage with the infectious disease folks? Was that hard, easy?
I'll just say, we have such amazing infectious disease clinicians here. Quite honestly, when I came here, you know, for a postdoc, I think that's where I really learned how to do collaborative science. I learned it on this amazing campus, and I learned it from the infectious disease person who were welcoming and open and supportive and just great work.
You know, Victor Nazay was really active all across the campus, and he was really, you know, our first and our longstanding collaborator in the clinical world. And then George, who's a very creative infectious disease doc, also joined the team. So, yeah. This campus is truly collaborative and interdisciplinary.
Hi, this is wonderful, wonderful presentation. At Vision's first symposium here, there was an investor who gave an address saying, I look around here and I see all the cranes and I see all the buildings, and I think, what a misuse of resources. Your administration wants to focus on buildings, but there are billions of dollars worth of IP lurking in the labs in this university that aren't being commercialized or explored. And you have done the exact right thing, and yet you're still struggling for money. So I'd like to see you back here and say, well, where's your money now that you have a company that has proven itself?
That's great, yeah. Do you have anything to add to that one? Yeah, well, the company is still, I would say we're not exactly struggling for money, but anytime you have a CRO business, it is a little stressful when you get the next contract. But the company, it continues to be profitable. So it's doing really well. It's a small, streamlined operation, but yes, I agree that UCSD is top 5 in many biomedical fields, right? I mean, it's top 5, but where are we, for startup and venture capital investment? So to me, that says that we have some gain to make in this biomedical research space, and I think this is something where we could do better at UC San Diego, because we have everything. To me, that delta between the top 5 rankings and the not, depending on where you put us, not at that same level for sure, is an opportunity for commercialization. So I think that's absolutely correct. Thank you.
I wanted to ask you, do you think your academic research has benefited from the commercial side of things? Has it opened up new avenues of research or anything?
It's interesting. You would think that, getting that company started actually took a lot of time and effort, and weekends and things. And my science just got better. Really got better. I felt like I was at the leading edge of what was going on in the antibiotic discovery world, because I've worked with most companies in North America and Europe in terms of projects. And so I kind of know what's going on all over the world. So I think it just benefits. More information, more knowledge is just beneficial to your academic research. Not that we apply that information, but it just keeps us focused on the cutting edge.
I guess I will only add to that Joe and I were both funded by the National Institute of Health, the General Medical Sciences. And the NIH funds basic research, but with the expectation that it will ultimately improve human health, right? That's what it's for. And so I felt it was really our responsibility to try to get this technology out into the market.
Please join us. Yes, I think we're gonna move on to the next speaker, but there will be more opportunities to hear from Kit and Joe, because that's the whole point. It's the lower barriers and taking cuts in the water. I was neglectful. I also wanted to just give a shout out to Stephanie Allen, who's our entrepreneur in residence, who's a great person in helping set this up. My colleague, Dr. Peter Suresh, who's an anesthesiologist and the Associate Dean for Simulation. And my colleague will be showing up at some point, Scott Mullaney, who's a nephrologist and has been very active in this space as well. So we're now going to move on to 2 flash presentations. The first is from Dr. Sierra Simpson, who is a post-PhD student here and now a scientist and has spun off her own company, Brilliant Biology. She's going to tell us her story.
Is that okay? Can everyone hear me? Okay. Thank you for that wonderful introduction. I am very happy to be here to kind of tell you a little bit about our journey. I started out as a non-traditional PhD student. I took some time between undergrad and graduate school where I worked as the first employee at a Stanford startup company, and that's really when the startup bug bit me. So I knew coming into my PhD that I wanted to be able to commercialize something. And my PhD was really at the intersection of the gut microbiome and neuroscience. And from that, I formed Brilliant Biome. And what we do is we're a microbiome-based precision medicine platform aimed to eliminate the risk of opioid abuse from the inside out. as many of you know, and I think, Kit, your talk, I was like, this is an amazing talk, I'm really enjoying it. You teed up the microbe problem and solution to, we have over 100 trillion microbial cells that live in and on us, and we only have 30 trillion human cells. They represent over 8 million unique genes that do functions from birth, like teaching our immune system what is good, what is bad. And we only have 23,000 human genes. So I like to think, are we more microbes than we are human? Are we just their little spaceship that drives them around? And really through my PhD and studying the gut-brain axis, which is the bidirectional pathway between the gut and the brain, we learned that the microbes that live in your gut really can contribute to your physical and mental health. And I applied this to the opioid crisis, which we know is still a major problem with over 150 deaths per day. This increased 40% from the time of COVID. And we know current therapeutics don't work well. There's over an 80% relapse rate. So a one-size-fits-all approach is not working. And I think, we know opioids are a really important clinical tool for pain management. So we still need to be able to use them, but can we use them in a safer way? So our solution to this was actually leveraging microbiome data and artificial intelligence to determine someone's addiction risk before they were actually exposed to them. We tested this out in a preclinical setting of oxycodone self-administration in a rodent model. So you can teach rats to do anything. We implanted ports into them. You stick them in a box, you hook them up, they give themselves drugs. So they're actually making the choice to take the drug. And we did this in a heterogeneous outbred stock of rats. So they have the genetic variety that we see in the human population. So some really don't like drug. Some will sit there and they'll press 1,000 times for one reward. It's crazy. And so after I finished my PhD, we discovered that there were microbial signatures that were related to these animals that would go on to escalate their use. I really wanted to commercialize this and jump to the clinic. And I was looking around UCSD at all of the potential avenues that I could use. Coming from the Stanford startup company, I knew BioX was up there. And so they had relatively different kinds of accelerators and programs. And I was really happy to see that there was an entire incubation space essentially for these nascent ventures, anywhere from being a PhD student all the way up to faculty. And so I started out with the mini and micro MBA class. And basically it gives you a crash course in finance, founding a company, customer discovery, thinking about what is your business plan, get together a pitch deck, talk to your customers, understand how your solution is actually going to make it to people so that you can find the most feasible way to get there. And then we were accepted into the StartR Accelerator through the Rady School. From there, we built our pitch deck. We pitched at a bunch of pitch competitions. That gave us the first money to incorporate the company, actually. We won a variety of prizes through the Fowler Social Global Innovation Challenge. And then we moved on to the Institute for the Global Entrepreneur. They also have a program for acceleration. So again, it's like the customer discovery, and they use NIH I4 to help fund that, so that each little point that we took in this journey, I was learning, how do I sell myself for funding? How do I get my point across? What do I have, a 15-second, a 30-second, a 2-minute, a 10-minute talk? You have to break down all of these bites and know when you need to launch them in order to get your company's perspective across. We also received funding from the UC Startup Innovation Challenge. We received one of their social innovation awards. So that was another, $10K. And 10K adds up, especially when you're a PhD student and you're like, I'm making no money. How do I start a company? Really, it was these pitch competitions. And during COVID, when we founded the company, everything was virtual, which was actually perfect for us because we didn't have to pay to fly to a place, stay there, hope that we actually were going to win some money. Otherwise, I would just be out the money to get there. I like the idea of actually keeping them virtual because then it really adds to accessibility. Then we wrote an AIM grant, which is the Accelerating Innovations to Market. And we were awarded, I think it was $47,000. And that actually was the key funding to get us into the clinic. we were connected with the Device Accelerator Center at ACTRI, and I started working with Dr. Rodney Gabriel and Dr. Ruth Waterman, and they really enabled me to move from what I would say is like a preclinical researcher to seeing the backside of clinical work, how we need support from clinical coordinators, what is the process. You know, I've written a million, animal protocols, and I know that whole process, but writing an IRB and making sure that we were, I was trained from City and I had all of my back end things that I needed to work with human data. I wouldn't have been able to do that without that initial ACTRI support, and then moving from my home department in psychiatry to anesthesiology. And so we moved into clinical validation, and we started a study. We were able to recruit almost 100 individuals, where we were looking at their microbiome before and 12 weeks after joint arthroplasty, which has a higher rate of opioid dependence afterwards. Turns out our surgeons are really good here, and so the rate of people continuing opioid use was actually quite low, but we got some really key data that will enable us to move forward with that program. So it was really exceptional. Some of the other ways that we've used tools to bring in and bootstrap, because I think, Kit, you said, don't take the VC money. I also did not want to take the VC money. We, of course, was the Fowler Social Global Innovation Challenge. We won second place for Amazon's Startup Challenge, and I think it was 2021. They had over 10,000 applications. Again, virtual and amazing. And then our most recent thing that I'm really excited about is that Google actually brought us into their AI and ML accelerator and gave us $350K in computational services. And since we're mostly machine learning, that's where our spend is. So for the next year, we're good to try out all the things that we wanted to try with really large data. And they've been exceptional in the support to moving to GCP. I would say, we spent some time with Amazon, which was also amazing. But I think that we're really now ready to scale with Google, and we're creating deals that are also bringing in a scale of data that I was not ready for, but I'm excited for. So I'll take any questions from there.
Thank you.
Very cool. As someone who has no experience with any kind of clinical trials or work, I hear numbers like $1 billion, $100 million, and it makes it hard to imagine what you can do with $40,000. So if you could elaborate on how that money is to be spent, that would be really helpful.
I've got one. Hello? I'm frugal. Most people would say I'm pretty cheap, which I think is a good thing when you're starting a company. With that $47K, basically what it covered was making all of the kits that we would need to collect the feces for that study. It supported the clinical coordination services, so finding the patients, consenting them, getting them the kits, getting the kits returned to us. And the biggest cost for us is sequencing. So we do shotgun metagenomic sequencing. It's at the core here at UCSD, which I would say please take advantage of the core services. It's like half the price that you would pay to have another company do it, and they're really cool people, and they're very willing to work with you on like what you need. That was about 25K of that 47K, but it enabled us to sequence 100 people to get a sample size to compare to the preclinical work that I had done and find our first hits.
Hi, how you doing? Thanks for the presentation. I have a question a bit about the technology and sort of where ideally you'd like to see it going. And maybe I'll ask this question in a leading way. It sounds like you work obviously with clinicians to gather the samples, et cetera. Do you see this ever being sort of a home kit that people can utilize? I've seen similar things looking at urine where it's actually in the toilet itself. So I'm kind of curious how you're thinking about that.
Yeah, so we have both the B2C and B2B channel. we do, patient stratification for clinical trials, but there is also a home kit that is available. We also white label our kits for support for other diagnostics. Some of those like early commercial B2C type kits actually helped pay some of the bills when we were getting started. It wasn't a lot, but it was enough to give us some discretionary funding to, do some things that we needed to do. But dealing with the public is entirely different than dealing with a big company.
Thank you for sharing your story. It's really interesting, especially having no background in this and coming from the clinical research side. There are two questions. One, on the clinical research, you have to show in a smaller group a signal and then ask for money to do a bigger study. So how did you go around about looking for funding with how much data did you have to show that this might work? And the second is, all these the grants that you got or the Google or Amazon that you work with, how much of the piece of action did they want and how much did they sort of have a say in what you did with the money?
So for, say, Amazon and Google, they actually didn't take anything. These are to support development of small companies. They want you to buy into their atmosphere because you're going to spend a lot and compete with them if things go well. So it's a minor investment for them to support, small businesses. And then, sorry, what was your other question?
The first one was, how much data did you have to show that this is a good thing to put money in?
This is also a testament to my cheapness. So we built a mini PubMed library, and so we scraped all of the PubMed data and used that as a tool to validate all of the hits that we were seeing in a variety of our platforms, and then also our preclinical data to validate that. And that was enough for some SBIR applications. I wouldn't knock also looking at government funding like ARPA-H. They're very targeted, but if you can flip how you write a grant, it's very different than an NIH style grant. It's very technical. They just want milestones. Those can be quite good, in the $10 million range. And then, I think I'm always talking to people, and then you never know who's going to say, hey, I know this person in the government who would be interested in looking at opioid use in the warfighter. And so I've just tried to leverage all of those connections by saying, this is what we're doing, how can we collaborate? And I love to collaborate, so if anybody's interested in the technology, I'm more than happy to talk to you. Like, I can't wait to talk to you and Joe, because I have some cool ideas and I'm like, ooh, this will be a fun collaboration. I think it can be daunting to go onto a website and be like, what kind of grant can I get for the commercialization of this? But SBIR is a good place to start. And like I mentioned, like Eva Garland has a program for you so that you can learn how to write them. And they basically write your first one for you. I didn't need that help, but it was really nice to have.
Hi, great presentation. I was just curious, so you mentioned that you're developing B2B and B2C type relationships. Could you say, I know this is probably a big question, but in terms of regulatory, what kind of resources are you thinking about? Because eventually that's where you're going to have to, thread the needle there as well.
Luckily for us, the microbiome and machine learning are both a little bit of the Wild West when it comes to the FDA. And I've navigated this process before with AAVs when we were first looking at putting viruses into humans. So I have a lot of experience contacting the FDA, initiating these pre-IND meetings where you say, hey, like we might have this real world data. Can we use this for a potential future filing? Which pathway would you like for us to have? Don't be afraid to reach out to the FDA and ask them about your technology. It's better to do it early so that you can use your research dollars wisely to get to the endpoint that they want to see.
Would you kindly share your story about how you found out about the micro mini MDA, how you went about doing that, what cost, if any, you incurred, and how long it took?
Yeah, it was a while ago. I think, what was it, 2018-ish? I've been around for a while. They were advertising it through our Lunch and Learns as graduate students, and I think it was $900. My boss paid for mine. He was more than happy. He's very supportive of all of these efforts, actually, from my PhD. They also, I think you had some scholarships at that point. There was like some flexibility in helping graduate students actually get access to it. And then it was.
Let me just add that the micro MBA is very economical. It's like $250. I actually started this program about 10 years ago. It's mainly it's an entry-level program for postdocs, PhD students here on campus. And the mini MBA is a partnership with the medical school here. And we also offered it as a part of the engineering school, but it's mainly the medical school called BEAM, the program that Charlie, Preetham, and we're all involved in that. And we can get more word out to you. Okay. So it's different price for trainees, residents and fellows, highly subsidized versus faculty.
Yeah.
And how long did it take?
I think
The micro MBA is like summer, 4
yeah.
weeks in the summer. And a mini MBA is also 4 weeks in the fall.
but
Okay.
much more intensive.
Okay, thank you.
Thank you for that excellent talk, Dr. Simpson. And I'm going to ask you the same question I asked Dean Poliano, but I have to declare some conflicts here. So I am the guy who worked with Dr. Simpson on the licensing and getting the company initiated. I was also on the review committee for the Accelerating Innovations to Market panel.
I didn't know that.
Yeah, you didn't know that. That's how it's supposed to be. And don't spare my feelings, but same question to the Dean. How did you find the licensing process, particularly also the AIM, which was new, that didn't exist back then, and the journey into things like SBIR and living funding?
Yeah, I mean, we wouldn't be where we were without AIM. Really, AIM was a transitional point for us in terms of looking for investment. We ended up taking one investment from one individual to kind of expand some of our options. But in terms of licensing, you made it really, I mean, it was an open conversation, which I thought was really helpful in that whole process. And it was pretty challenging because our technology was distributed across UCSD and Scripps, which once you get multiple institutions, it can be hard, but Chris made it really easy. I think also in terms of licensing, you were doing your best to make it feasible for us to be able to handle the license, and I thought it was really fair. So yeah, so the turnaround time was great. I was very happy with it, and it really helped give us credibility when we were out pitching and meeting the community, especially as a graduate student. Thank you.
Why don't we, Sarah, thank you so much. We'll switch our seat here. So we'll stop there. I guess I love the questions because in my mind it's what would it take to get my face up here? There's a lot less friction to this than you might imagine. There are a lot of resources, and all the people speaking will be available either later at lunch, and there'll be these guys will have a little bit of a panel discussion afterwards. So next, it's my pleasure to introduce Dr. Steve Moyo, who's a hospital medicine clinician here at UCSD, and himself an entrepreneur who will be talking about his company called Wellfee. While we have this little gap, there was another question, or you want to put it out now?
Yeah, maybe we can take Uri's question for Sierra.
I have a nerdy question for Stephanie.
Sierra.
Oh, sorry. Nerdy science question. What do you think is the mechanism by which there's a relationship between the gut microbiome and susceptibility to either addiction generally or opioid addiction?
There are three main pathways for the gut-brain axis. One of them is the vagus nerve, which can actually sense like interoception in the gut and how that's interfacing with the microbes. Two Microbes release metabolites, and those metabolites can signal both peripherally and centrally. And then 3, through the hypothalamic pituitary axis, so stress regulation can also change the makeup of the microbiome and how that feedback is also altering.
Follow-up question.
Yeah.
Which metabolites do you think are,
We have hit lists of our favorite metabolites. I mean, some that you probably see in the news are things like short chain fatty acids, bile acids, even local neurotransmitter regulation is really interesting. We know some microbes actually eat those transmitters or produce them, so it can cause inflammation or dysregulation in gut in general. It's still being determined whether or not those peripherally made neurotransmitters act centrally, but we're figuring it out.
cool,
Great, that's gonna be super interesting. All right, I think Stephen, you ready to go?
Yep, I'm just gonna say like one thing to make it a little bit easier for me, so I'll keep it on my head. 1 second. No, I'll just write to the thing not do that. Hello everybody, I'm just gonna hold this. Pleasure to be here today. I'm Dr. Stephen Moyo, CEO and founder of Wealthy. Wealthy is a health equity platform. We're building software really to help community health leaders care for and connect with their communities. We particularly focus actually on families that are underserved and families with young kids. And our goal is to really empower healthcare providers in the school setting as well as in the community to better deliver health education, increase access to care, and really start to build the community health workforce that we need. I started this journey about 5 years ago, and I'm gonna, at the back end of this presentation, sort of go into that a little bit more. But what I wanted to do was really think about those of you who are in the room who are thinking about taking this journey, and give you some of the information that I wish I had known and some of the stuff that I've learned along the way. And really what I want to present is a bit of a framework. So along the way, I've come up with this framework, and as I talk to entrepreneurs who are thinking about starting entrepreneurship, this is what has helped me sort of communicate to them sort of where they're at in their journey. But also as a company, as we grow and we innovate and think of new products, this is something that we're putting into our company as a framework to say, where is this in our roadmap, and what do we need to do to get it sort of through the line? So the D's that I like to call them are dream, discover, design, develop, as well as drum, and there's a 7th one that we'll talk about later. Some of us here are clinical, so this is the way you can think about it. In high school, you're dreaming about becoming a doctor. In undergrad, you're discovering what it really means to go into medicine. In medical school, you're designing a physician that you want to be. In residency, you're developing all those skills in a really high iterative way. You start to deploy your minimal viable product in fellowship, and then ultimately as an attending, you sort of perfect, you optimize the clinical practice that you have. So, to dream. I think this is important. This is what a lot of us do, and we have that internal voice sometimes when we have an idea that says, can I really do this? Do I have the time? What's it gonna take? I think it's really important that people allow themselves to do this and have the chance to do it. One of the key things that enabled me to start my journey was the transition in the time that I had. So you go through residency and you don't have any time for yourself. And then as an attending, I work as a hospitalist here. I started to have a lot of time on my hands. And so I took that time to think about the problems that I wanted to solve in the world. After you get through residency and you have your career starting, don't get me wrong, you're busy, but you have a little bit more time to think about what you want to do with your career, or frankly, what you want to do with your life, because you spent the past decade focused on getting this degree, this doctorate, and this career started. The next piece is about discovery, and we heard a little bit about it here. It's really just talking to people. It's taking that internal idea and externalizing it, and just getting it batted around a bit. I love the phrase, which is everybody has a plan until they get punched in the face. That's kind of what discovery is, and it's okay, it's really essential. Because one, it's gonna test your conviction about the dream that you had, right? Can it take a few punches and can you really stand up and back it up? 2, you just start to get it socialized. Start with family and friends, right? Find those people who are champions, and find those people that are also really good, like positive naysayers, right? That could be anybody that you might know. It could be your mom, it could be your wife, it could be your husband, it could be whoever it might be. But that person's actually really valuable because they're gonna poke holes, and you want people to do that at this point. Because when those holes get poked, you can figure out how to move forward with it. The next piece that I think was integral, and I want to ask this, I didn't do all of these in order, this is sort of after the fact. Put them in order, and why I'm putting it in order here. for you if you can do it this way, but it doesn't have to be done in order. But this design process is actually critical, right? So we have a software product, and designing that process, going through the design process with the designer that is doing human-centered design. We've done it both with and without, and I can tell you the outcome when we do it with is always better. It's slower at first, it's more intentional at first, but it moves faster downstream, and you have happier customers. The next part is developing, that's getting people in front of keyboards and typing out that code, that software, actually building the product, that product's doing it fast, doing it quickly. And then ultimately deploying it. One of the key things that I've seen a lot of founders and innovators do, they get stuck in the design and development phase, right? It's really easy, because the design and development stage is actually very similar to the dream stage, it's kind of internal, right? You might have a bit of a bigger team, but you're building this product, it's not really out there in the world, so nobody's punching it in the face. And it's only once you actually deploy it and get it out there, and you really start to learn that you build the right thing, and really trying to get through these as fast as possible. If you can have some of these things happen in parallel is really key. And the last part is what I like to call a drum. So if any of you have ever played in a band, and I did it back in the day, the drummer is actually the most important person in the band as far as I'm concerned. I'm saying that not as a drummer, but as the frontman. The drummer is always listening, they're always adapting to what the bassist is doing, what other players are doing, what the frontman is doing. They know when to speed up, slow down, but even when to stop. And what I really mean by drum is about iteration, right? It's about listening to your customer, it's about getting better and better. In Japanese, I believe there's a word called kaizen, which is continuous development or continuous improvement. And I really feel that that's what the key part is here. So with that, I'm going to talk about the seventh hidden D, and that's when do you dive in? When do you really commit to this innovation process? It's going to be different. Some of the people might do it at the dream stage. Some people might do it when they finally deploy and we're seeing customers really take this up. For me, I decided to really dive in after the design stage. After we got back the first designs and it was tangible and it looked professional and it wasn't sort of my scribbles on a piece of paper, that was when I really decided to dive in and say, all right, I'm going to do this. We're going to do it for this amount of years. We're going to raise this money. We're going to do X, Y, Z. And so with that, I want to take you once again through this process, but through the lens of my company, Wealthy. What I'll actually do is talk about it. I mentioned we use this internally. If you're not a startup these days if you don't have AI in your company name or you're not doing something with AI. AI was actually something that we wanted to do very early on in our process when I founded the company. But the AI that we talk about today is very different. It shows how quickly the field moves. And so we've had to sort of redesign and redevelop and rethink how we're going to bring this new type of AI, generative AI, the tools that we have readily available into our company. And it's really changed the way that the product's roadmap looks like. So I'm going to walk through this. For me, as we talk about this specifically, the dream around generative AI, I took kind of a year to understand what was going on with, ChatGPT. Was this really just another thing, the technical thing we were really excited about and go away? Was it really a clinical application? So for me, this dream actually started looking at the work that my wife does. She's a clinical psychologist, and she cares for a lot of kids in this community. And if you know any psychologists, they spend an inordinate amount of time writing notes. Some of you are clinicians here, so if you think about our psychiatrists in the hospitals, the same thing. They have the longest, most accurate usually, and really fantastic notes, and they often get copied, forwarded, and pasted into everybody else's. But as we think about that kid that's in the community who needs to see a psychologist, they're actually less likely to see a psychologist because that psychologist is spending 4 to 6 hours writing an assessment. And so a lot of psychologists are actually moving into doing therapy and away from assessments, creating a bottleneck because it takes so much time. So the dream was simply how can we create and use generative AI to make the writing time shorter for pediatric psychologists so kids can get increased access to care. So I did discovery. I talked to my wife about it. She said, there's an AI there, like it can help her. She also talked to her colleagues. And I talked to some of my friends who are colleagues as well. This is Dr. Shannon Scratch. I actually went to high school with Shannon. She's a very dear friend of mine. But the reason I went and asked Shannon is because I went to high school with her. She's really easily ready to punch me in the face, right? So she's really easy for me to talk to. She'll tell me right away, Steve, this is a horrible idea, don't do this, or there's an error there. And this is one quote that she said that I think really took away the why that I thought we should do this, right? The time I say writing, I can spend thinking about or spending more time with my patients. So for her, it wasn't even like I can see more patients, is I can sit down with my patients and give them better feedback and give them better care. And so it was intriguing to learn the different reasons why people thought this technology had legs. So then we went into rapid design. A short story about my first design experience. I mentioned my short scribbles on a piece of paper. The cue card was sort of my best friend when I started this journey. It's the perfect size to think about like a mobile phone. You can sort of sketch out your app ideas and move them around. But it's a really humbling experience because when I first called up a developer, I was like, all right, I want to build an app. They're like, okay, send me your wireframes. I was like, what are those? And I didn't say that right away. I kind of like, BS it for 10 minutes. They're like, yeah. And then I was like, eventually I was just like, all right, what's a wireframe? So they told me sort of some software. I think the first one I used was Envision. You can use Figma. Frankly, you can literally just use PowerPoint. But what a wireframe is sort of the initial, very basic design. I've gotten a lot better at it now, but this was the next stage we went into, rapid design. And I was able to show this to Shannon, show this to my wife, show this to other colleagues, create videos, send it out, and just start to get feedback, right? Is there something here? Do you not like it? And we have some people who don't like it and say, I don't think this is a good idea. And I'm hungry to talk to those people because if we can't convince them, we might not have somewhere to go here, right? So that design is really important. And then we start to develop it. And so this is actually something my CTO sent me just last night. He's been hammering away at the keys, sort of building this out. This is real time, so we haven't sped it up or don't have enough GPUs right now. So all the things you mentioned about compute power, we're sort of at that bottleneck. But the real question was, can we start to build something that could generate a clinical note that would decrease the time for a clinician? And so I'm not going to let it go through the whole thing, but you'll see it start to build here. But on this point, I think what the key thing about this story is sort of rapid iteration. We went from dreaming to designing, discovery, developing very, very quickly. And now we have something where we can start to show, start to pitch, and really start to try to frankly raise some money as we think about deploying this. We haven't deployed it yet, right? And that's because we need to be really intentional and really safe in healthcare. But one of the reasons we dove in to do this is because we can deploy it in a very safe way, which is we have clinicians that we know who can sit down and write notes and we can give them on patients. And if we either give them a little equity, whether it's sweat equity or equity in our company, or just call on some friends, we can actually start to get this deployed quickly and sort of iterate. And last but not least, how do we get better? Now, I mentioned none of this stuff has to be done in series, it can be done in parallel. One of the things we learned immediately when we started talking to customers, potential customers about this was, oh, it would be really great after this note if you could help us build, right? Or if you could help us build faster and get this money back in. And so already this is that drumming, this is that iteration we're listening, where we're putting this into our roadmap at an earlier point, because pretty much every psychologist we talk to, this was a pain point. They're using spreadsheets to track how they bill and how they get the money back. So that is it. I'm ending with this, which is my favorite creator right now, Donald Glover's got a great show on Amazon, which is just to stay creative. I think being a founder is all about creativity, right? But it goes back to that dream moment and having the creativity to think about what the world could look like, what a better world could look like. So stay true to your creativity. So with that, I'll take from that.
Thanks so much, Steve. Maybe we'll open it up to everyone here and you can ask questions whoever you want for the next bit, and then we'll take a break at about 10:30 or so.
Why don't you all come up with you'll have
Yeah.
Joe, questions?
Yeah, go ahead.
There, this question is actually for everyone on that presentation. And I'm Tony Olbert, I'm a president training at UCSD, so that's the perspective on this question. And I'm curious to hear, if you launching a venture with sort of one of your aims being health equity, how if you've grappled with sort of the form that venture should take, with wanting to do good and sort of increase equity in the world versus being sort of a profit-seeking enterprise, and what the best form for that sort of enterprise or venture is, and how you sort of marry those two perspectives.
Yeah, that's a great question. We ask that ourselves all the time. So when I started it really was with the history and experience that I started charities before. And charities can be great vehicles for change. I think when they're first starting, what I realized was it's really hard to attract really talented people. You can do it, but not a lot are willing to sort of stop their careers or completely leave their careers. And then the other part of it was really just thinking about where I was as an individual. I think as a physician, as a clinician, you get to have a positive impact on the world. And so that kind of checked the box for me already. So the question is, I want to do this at scale. What is the thing that I think is going to make it grow fast, grow to a very large scale, but then also be sustainable? I haven't, at that point, and I'm not an expert on sort of charities, I haven't seen a lot of sustainability models in charities. It's keep asking for money, and keep showing impact. Where with capitalism, frankly, you're really trying to build an engine to spin off cash, right? And that cash can be reinvested back in the company, it can go to your shareholders, it can go to you yourself, it can go to motivate your employees, right? And so for me, it was, okay, set the mission, stay true to the mission, but also chase mission and margin at the same time. And then I think the last piece I'll mention is there's actually companies that are mission-led and purpose-driven that outperform. And so to me, it was kind of, this is actually the best capitalistic decision we can make as a company, is to be mission and purpose-driven. And I think it's proven out in the people that we work with and the people that we're able to attract. We have phenomenally talented people who are willing to work with us, whether it's full-time, part-time, or just help, because of the purpose and mission of our company. And they've said, like, if you guys were just here to make a buck, I would not be offering you this. It's because I believe in the mission that you have. And so I really do see it as a competitive advantage. And if that is, something you're interested in, hold to that mission and make it the first thing you talk about when you walk through the door.
Great, thanks.
Hi, my name is Luz. I have a question for Sarah. I'm a microbiologist. Well, I was a long time ago. Then I, transitioned into immunotherapy and cell therapy. So, I have a very soft heart for microbiology. I know how complex it is to, find hits in the microbiome. So I'm just curious to know, once you identify this differences between people that have this susceptibility to become addicted or not, how do you think you will apply this information? Because people are gonna need pain medicine, right? Anyway, so it's like if I'm susceptible to becoming addict, that's one thing. And the other thing is, and I hope the industry and healthcare is doing this, I had a surgery recently and I was being bombarded with opiates. I didn't even need them. They kept pushing them onto me and I'm like, I don't really have. So I was very impressed and I could hear even other patients in the room, the nurses, it was amazing. I was just shocked by how much they pushed the opioids into people. So this, it's a little bit concerning.
Yeah, I mean, opioids are a really important clinical tool for, managing pain. So I do think that they are important. And I think, the public is becoming more educated about their use. So yes, very necessary. How we're going to get people to change so that we can actually enact change, right? Humans are bad at changing habits. We're like, oh, that may be good for us, but I'll do it next week. So the way that we want to approach those hits is not only using microbes, but also their metabolites so that we can act directly at the source. And then doing some medicinal chemistry to make it better. So eventually doing drug development and drug discovery.
So to clarify, you're discovering drugs from bacteria from patients who are less susceptible. Is that a fair summary? So patients who are less susceptible have bacteria that produce anti-addictive compounds. And you hope to find those drugs and then you can provide them to people who do not have that microbiome. That's really cool.
Hi, I guess this is for any and all of you, but I'm curious, as we're developing your business plans and talking to tech transfer offices, how did your, business acumen, develop? And did you do most of this by yourselves, or did you also then go out and seek help? Because oftentimes so I'm the Schneeberger, I'm at the Solid Institute of Technology Development, and we often find that there's a big knowledge gap. And you can go and do a mini or a micro MBA, but there's a lot more to it depending on which field that you're going into. So you guys have obviously been quite successful, so I'm curious how that developed.
I gotta pass the mic, so I'll start. So for me, when I decided to start this journey, I kind of had two paths, which was go into an MBA, which I think most doctors and after so much school is kind of the default. Like, I need to learn about this before I can do it. Then I kind of just had I think two epiphanies. Is one, it's not that high risk, right? So medicine is different. You need to go study it before you start prescribing medications and all X, Y, Z, people flags on the line. When you start a business, depending on what you're building now, right? But if you're a responsible person and you're not going to put a bad product in the market before it's too early. When you start a business, the only thing you're risking is a little bit of time, right? Perhaps a little bit of ego, right? When you tell people that you have an idea, and then a month or two later, like, what happened to that idea? You're like, yeah, it's not going well, or I killed it, but it's just done. But there's not a lot of that you're actually giving up. So for me, it was don't go with your default, which will delay you starting this business another 2 to 4 years. And what probably would have happened is I probably would have ended up working for somebody else, right, with that MBA degree, right? And so for me, it was jump in, fail, right, and keep failing. And when I learned that was a key part of entrepreneurship was to rapidly fail and learn from it, and embracing that made it a lot less scary.
I think you bring up a good point in kill it fast. You don't want to linger on something that is only kind of half baked. And in my PhD, everything fails for the first 4 years. And then you're like, oh, something works. I have data. This is amazing. And surrounding yourself with a good team. You don't necessarily, having the fundamentals by taking the micro and the mini MBA gave me an entry into what I needed, learning what I needed to know and what I didn't know. But you have to have a good team to support you. Being an entrepreneur isn't a solo thing. It's the community, really.
So we had no micro MBA, no business experience. Initially, I could barely use Excel. I was forced to get really good at it for accounting purposes. So two things. One is where we had really major gaps, we brought in some experts. So of course for legal, where we're going toe to toe with big companies, we had a really excellent lawyer. So we have that aspect being covered in terms of negotiations. In terms of just business practices, we decided to take the same approach to the business as we did to our science, which was to be creative and look for novel solutions. If there isn't an obvious solution out there, we'll try to invent something. And that's how we ended up, getting clients, putting in complex and unique partnerships to make it happen. In hindsight, maybe if we could have had a super experienced CEO come on, the company would have been a very different company. But I think, as you said, we would have been probably much less involved and probably, who knows, if it'd even still be in business. But by doing it all ourselves, we maintained 100% control of it, and it's still going and profitable and happy. I think if you saw on Chip's slides, all the employees basically came from UCSD, which is kind of nice.
Yeah. Great. Last question, then we'll just take a break.
Thank you all for sharing your entrepreneur journey. Everyone in this room has some sort of technical background, all experts in this room, and you've all identified some of the competency gaps and have identified the right people to help fill that. So one thing that I want to step back to is basically there's a tiny survival bias of the fact that in a room like this, most of us can remember when we were in undergrad and in grad school, and every person in there was extremely, really smart. but ends up really coming up with great ideas, but never following through. And I think one element, not all of the variables, but one element is really that shyness or the, shall we call it charisma and the like. So let's go into the soft skills. How did you individuals, on the panel really develop that? Did you think that was something that was innate? Did you really train for it? And what's the likelihood that, the remaining 98% of creative people in your undergraduate, graduate program, and probably, medical school and the like, may be able to get out of that shyness and come up with a great company like yours so that we can actually scale it and not make it more the survival bias of a charismatic technical scientist. Thank you.
I think for me, and so I guess I'll try to just speak from quote unquote the shy person's perspective, which I'm not. I'm not shy, but the reason I'm not shy and the reason I feel comfortable talking about my company is because, as I mentioned, it was a front end in the band, right? I got actually really comfortable behind a microphone and having this like idea, telling a bunch of people about it, which is the song, right? Having people work for a really short period of time to get the song done and then performing it. And so I realized very quickly, like, that's what pitching is every time that I do it. It's like putting together a story, having a team, and then do the front end. And so I mentioned that to say that most shy people I know have like this thing that they're really good at, really get excited about, and the shyness goes away. My wife's an introvert, but you get her talking about some things, and like she'll push me to the back of the room and be like, I'm gonna step on the soapbox now. So I think if you're that person, finding that intersection between your expertise and the thing that you're excited about, that is your comfort zone, is perhaps kind of the best way to do it. And then as was mentioned, team, right? If you're not the person to go out and pitch the company, but like, hey, I can build a product and do that really well, find somebody who's a great talker that can network and do all those pieces that a business needs. Because you tell them that's your job, I need you to raise money, sell the business, build the network, which is essential to the business, and I'm gonna build you a great product that will work. And that's the team that you need.
Yeah, just because you are the inventor of the thing that you're trying to commercialize doesn't mean you need to be the CEO. I have an amazing CTO who he loves to be behind the scenes, but when I ask him, I was like, can you ninja this into existence? And two days later, I'll have a prototype of that thing, but he would not want to be out here talking to people. And I'd say no one will believe this, but I am a pretty shy person, but it took many years of just like getting up in front of people and practicing. Teaching a class when you have an opportunity. We're at an academic institution. You can always look at who's teaching classes and say, hey, I'd love to fill in for this class. It's a good place to start. There's also Toastmasters. I know I didn't do it, but many people I know did. And that helps you get practice just talking about things that you're passionate about. And yeah, using that passion is really important in showing people who you are, and that becomes part of your brand too.
Well, so okay. So I guess, I agree with the teaching piece, right? We're both professors, and so we're used to getting up and doing the pitch in front of the students effectively, right? You try to get them excited about the things that you're teaching them, and that kind of gets you over any kind of, public speaking fear. So that's a really, really great suggestion. I consider myself an introvert. I know it's not really likely I'm a dean, but anyways, because I'm really passionate about the things that we do, right? At UC San Diego in the School of Biological Sciences, and also in Linnaeus and in our research program. So that's really, that passion can get you up and in front of an audience and feeling totally happy. And can make you forget you're an introvert. Did you want to add anything?
Let's go Gary's
I want to hear what Joe has to say too, but I have a follow-up question to this line of questioning.
Just briefly, everything they said was great. And if you have an idea, like for me, I don't think I'm that good of a speaker, but when I'm passionate about something, oh my God, I can't wait to share it with you.
Mm-hmm.
I'm so excited by it. And that is, I think anybody can tap into that. And if not, then you should get someone else to speak for you.
That's the perfect segue to my question. Can everyone hear me okay? I don't think I need a mic, okay. I have the flip side, which is that I get so passionate about ideas that I want to talk to anyone who will listen about it. And I'm surrounded by people who say, I can't talk about that. Whether it's an industry or it's in academia, people don't want to, because they're worried about IP or losing, or giving away secrets. So is there a formula or algorithm or secret sauce, or how do you know? Because if you don't talk about the idea, if you don't get into the details, you might be missing a chance to get some of the best advice from people. And signing a CDA and doing all of this stuff is just so slow, especially if you're at a conference or whatever. So does anyone have any advice on how to figure out if you're talking about something you shouldn't be talking about, sharing details or ideas you shouldn't be sharing? And how do you speak freely and passionately to get feedback?
I'll just answer really quick because I think we're running out of time. But as an entrepreneur in residence here at UCSD, I interface with a lot of faculty, and this is definitely a concern. We hear from all the panelists, go out and talk to people, talk about your idea, but there's a fear that they're giving away their secrets. But I always remember that, you can talk about it on a generalized level, enough to get your point across, but remember that there's a lot of ideas out there, but if you can't execute on it, it's not gonna go anywhere. And you have to have the team and the talent to be able to execute the idea. And someone else is probably also thinking about your idea in your space. So it can alleviate some fears.
So you're encouraging us to be open.
Absolutely.
Okay.
So let me just say that's a nice segue to actually the next panel with some experts on this topic. Okay, Chris, Richard, and Marshall. Okay, so let's take a quick break and let's have a coffee here. Let's thank the panel here. Thank you so much. Steven, see you.
Yes, some of them are.
Okay, good.
Oh, yeah, because right now,